The next speaker is Dr. Bruce McEwen from the Rockefeller University. The title is The Brain and Body on Stress, Epigenetics of Plasticity During the Life Course. Please, Bruce.
Thank you very much. It's such an important meeting, such an important topic, and I'm very much honored to be part of it. What I'm going to do is to do as Jack Schoenkoff says, a deeper dive into some of the biology and neurobiology that you've already heard about, having to do with how experience affects the brain and the body, and how the brain is affected in particular by early life experiences that include not only the ACEs, but also experiences related to poverty and deprivation. So the objectives of the talk are to talk about, as I said, the importance of social and physical environment over the life course, using the modern terminology for gene-environment interactions, epigenetics. I'll talk also about the reciprocal influences of the brain on the body and vice versa. Talk about the structural plasticity of brain architecture and its resilience or lack thereof. Talk about, again, a little bit more about the lasting impact of poverty and other forms of early life adversity on the brain and body, something you've already heard some about and we'll hear again from our next speakers. And then, of course, address this incredibly important topic, what can be done to ameliorate. And I certainly don't have answers, but I'll suggest some ways of thinking about it. So epigenetics is originally a term that really referred to how a frog egg and a human egg develop into different kinds of organisms. You couldn't have predicted from the one stage what was going to happen to the next stage. Now it talks about changing the expression of genes without changing the actual DNA sequence. And it's the modern term talking about gene-environment interactions. We no longer argue is it genes or environment. It's the seamless interaction of the two upon each other. And this includes, for those in the biology, mechanisms such as the modification of histones, the binding of transcription factors, the methylation of cytosine bases in DNA without changing the genetic code, non-coding so-called microRNAs that regulate our messenger RNA survival and translation, the editing of RNA after it's transcribed, and then in a somewhat different category, the jumping genes idea of Barbara McClintock, the rearrangement of DNA, which does change, the actual sequence of genes within the chromosome and their implications for function. And I obviously won't really talk in depth about any of these things, but just to remind you what they are becoming, because there seem to be more and more examples, mechanisms. There's a PBS documentary, NOVA series on epigenetics, which is very useful if you're interested. You can find it on the web. The other really important notion having to do, it comes from this article by Neil Helfand at UCLA, talking about the life course health development perspective, in which he uses sort of computer language to talk about 1.0 operating system, which was finding penicillin. You could say nowadays doing open heart surgery to help people survive from cardiovascular disease. And then 2.0 really goes back to George Engel in 1977, is what we're in the midst of now, the biopsychosocial model, in which we're concerned about the non-communicable diseases, obesity, cardiovascular disease, where the social environment and where health behaviors are very important, and how do we change these health behaviors to help us live healthier lives. What we've heard already today and what is abundantly evident is that it's early life events that have a disproportionate influence on the propensity for these later disorders. And so now we're talking about this continuum over the life course where you can't turn back the clock, and we have to think about how we direct or redirect the brain and the body at different stages of the life course. I'm going to talk about this figure from the New England Journal article in 1998 that introduced the notion of the allostatic load, but the notion that the same mediators that we use to adapt, like cortisol and adrenaline, immune system and metabolic mediators, can also cause problems if they are overused and dysregulated. And I'll take you very quickly through some of this so you see how the thinking goes. The basic idea, as I said, of allostasis is the active process of putting out mediators like cortisol that help maintain homeostasis, help the individual adapt. And this is their primary purpose. But obviously when there is a lot of challenges, whether we call them stressors or not, and when the balance of the mediators that respond is dysregulated. So there's too little cortisol, too much cortisol, too little inflammation, too much inflammation. Then this system produces a load which accumulates over time and is part of what we think are the processes that lead to these non-communicable diseases, where of course one's genetic predisposition and early life history are very important. We think of cortisol as an example. And one important thing to realize, of course, the cortisol is secreted by the adrenal cortex in response to signals emanating from the pituitary, which responds to signals from the hypothalamus, which responds to signals from physical and psychological stressors, challenges. But cortisol in the short run actually enhances adaptive immune function. It promotes memory of things that you need to know to survive if you escape the predator and so on, and this is a bad place to be. It promotes energy replenishment and more efficient cardiovascular function. But chronically, we know very well that it can suppress immune function. We use it to suppress inflammation. It impairs memory at high levels and intense levels of stress. And it promotes bone mineral loss, muscle wasting, and is part of the metabolic syndrome. So we talk about an inverted U-shaped dose response curve. Now, cortisol is not working alone. And in fact, it's working simultaneously with other mediators such as sympathetic but also parasympathetic activity, with pro and anti-inflammatory cytokines, with the other adrenal steroid DHEA, and with a little bit of oxidative stress, which is essential for cells to function normally. And concurrently, this network, and the arrows are intended to show that all of these mediator systems are influencing each other in a nonlinear network. It's concurrently affecting all of the organs and systems of the body, which is why there is comorbidity in many cases with the development of diseases related like depression or cardiovascular disease or diabetes related to allostatic load. So this network of allostasis, as we call it, can be dysregulated by an unhealthy lifestyle. I'll say more about that in a moment, including poor sleep, but also by toxic chemicals that feed into this network and, for example, promote inflammation, which then will lead to greater production of cortisol, dysregulation of the network. Lifestyle, very important, underappreciated when we talk about stress as fight or flight. because the daily grind of things that are happening to us every day in our daily lives is creating certain health behaviors. Whether we lose sleep at night because we feel stressed out, whether we eat too many of the wrong things, drink too much alcohol, smoke if we're so inclined, or neglect regular physical activity, which we know is good for us, but we may be stuck behind our computers or doing something that we have to get finished, and we don't do the walking or standing or moving that we know we should do. So psychosocial stressors are a very important part of what we generally refer to as stress, and they are contributors to allostatic load. So these are, of course, stressors, the trauma and abuse, major life events, but also the daily grind that I've just noted. Taking you through this notion of types of stress. Stress is an overused and misused word that means so many things, which is one of the reasons we think that allostasis and allostatic load is a better way to think about the whole picture. Positive stress, taking a chance, giving a talk, doing something with a bit of a risk, having a successful outcome, feeling rewarded. For this, you need to have a good sense of yourself, good self-esteem, good sense of mastery and control, so you don't take too big a risk. Then there's tolerable stress. Something bad happens, you still have these good qualities, you have external resources, both physical and emotional, and you can survive the storm. There will be some allostatic load that develops, but it doesn't go to the extreme that we have now called allostatic overload. And then toxic stress. And really what we're talking about with the progression of these levels of stress is greater or lesser and lesser senses of control over one's life. So there may, with toxic stress, there may not only be a lot of events in a person's life, but also what we call unhealthy brain architecture because of the early life experiences that we'll talk about shortly. But we mustn't forget that to include factors such as loneliness, circadian disruption, the ugliness, noise, and lack of green space in living environments, which also gets under the skin and affects the same physiologic systems and the same systems in the brain, albeit perhaps in somewhat different ways, but nevertheless, the overall result is a negative one. So our work mainly concerns the brain as a primary target of stress, and I can't possibly tell you as much as I would like to, but we deal with three brain areas we've already heard about: the hippocampus, which is important in memory, but also emotional regulation, but it also helps to shut off the stress response. We have the amygdala, which is important for emotions, fear, and anxiety. It helps to turn on the stress hormone response and also the autonomic nervous system response. And the prefrontal cortex, which is important for all manner of self-regulatory behaviors, for decision-making, for working memory, mood and impulse control. And it also helps to shut off both the autonomic and HPA stress response. So if there are changes in the architecture of these brain areas and their relative relationship to each other, because they all communicate with each other, then there will be disruptions not only in behavior, but also in the physiologic responses throughout the body. Over the last 20 or 30 years, we gradually recognized that the adult brain, as well as the developing brain, and even the aging brain, is capable of changes in dendrites. They can either shrink or grow, become less branched or more branched as a result of experience. That synapses in the middle, the excitatory spine synapses, that some of them are turning over regularly, and they're turnover can be influenced by experiences both positive and negative. Connor Liston, a colleague in Cornell, Weill Cornell, has shown that the ultradian fluctuations in cortisol are required for the regular turnover of synapses throughout the cerebral cortex, almost anywhere he's able to look in the brain. And this occurs according to a circadian cycle. And if you disrupt this, for example, with a tiny dose of dexamethasone that stops the cortisol, this turnover stops. And if you disrupt it too much, motor learning is impaired. So it's very important for adaptation on a daily, even hourly basis. There is a limited amount of neurogenesis that has been found in the dentate gyrus of the hippocampal formation, possibly in other brain regions, but that is subject to increases or decreases. Exercise increases it, certain kinds of chronic stress will decrease it, and the volume of that part of the hippocampus can either grow or shrink. Now, mediators, we have focused a lot of our work on cortisol, but we fully recognize that it works together with other mediators such as glutamate, brain-derived neurotropic factor, tissue plasminogen activator, CRF, corticotropin releasing factor, endocannabinoids, and there are others. But the other important thing is that while initially steroid hormones in the 1960s were identified as working through cell nuclear receptors to affect gene expression, this is still very much true. What this slide shows is that glucocorticoid receptors also go into mitochondria, and they regulate mitochondrial gene expression in a biphasic way, inverted U again. Glucocorticoids act directly on receptors that are like those for intracellular actions that are associated with the membrane to stimulate glutamate release, excitatory transmitter, but at the same time, they are working to enhance the production of endocannabinoids, which have the ability to inhibit. They go from the postsynaptic side to the presynaptic side, and they inhibit the release of glutamate, or in some cases, they inhibit the release of GABA. So they have different consequences for excitation. So this is just an evolving story and tells us that the mechanisms are sophisticated, complicated, and there's kind of a checks and balances that's built in to how how cortisol works. And not all is bad, since often we're told about cortisol in the in the context of negative events. We wouldn't be alive if we didn't have the ability to produce cortisol or adrenaline and these other mediators. So back to the life course health development idea, and the whole idea that individual experiences throughout the life course interact epigenetically with our genetic constitution and with the experiences that we have ongoing as we age. The idea that there are, from people like Tom Boyce, reactive or context-sensitive alleles that make individuals more sensitive to both positive and negative events, and that there are epigenetic modifications, even ones that can be carried transgenerationally, like perhaps obesity, some transgenerational modification of DNA in the sperm, that leads then several generations down the road to increased likelihood of obesity, and then behavioral transmission, which I'll talk about momentarily. We heard a little bit about individual differences, and one amazing thing, if you take a population of rats or mice from a supplier, they're supposedly fairly genetically homogeneous, you assume they are, but a study by Cavagelli and McClintock in the 2003 showed that if you screen adolescent rats from a colony for either their anxiousness in an open field, you take the more anxious animals and the less anxious animals, you follow them throughout their life course. The more anxious animals, both males and females, died on the average of 200 days sooner. So something is happening, and we really don't know what that is. One possibility, of course, from the studies that were started by Victor Denenberg and Seymour Levine and carried to important new heights by Michael Meany, Moshe Ziff, and their colleagues, is the whole idea that maternal care is very important. You can take pups from a not so good mom, it doesn't do a lot of licking and grooming of the pups, put it with a better mom, and those pups will show less emotionality. The females will become better moms themselves. Cross-fostering shows that this is possible, but also noting in the lower right of the slide, work by colleague Acacia Tang has shown, and not surprisingly, that it's not the amount of maternal care over time, but it's the timing of that maternal care and the consistency over time, especially in relation to when the pup has been exposed to something outside the nest and needs to have some comfort from the mother. So consistency is a very important factor. Now, back to the hippocampus, which has been our main area that we've studied. The ability, I'll just review for you, neurogenesis occurs in the dentate gyrus, and it can be upregulated by exercise, downregulated by stressors. It innervates the CA3 region, where the big white arrow is, and the apical dendrites of those neurons will shrink with repeated stress. But this is not brain damage. The basal dendrites don't change, and in a healthy brain, they will grow back when the stress is over. But they are not the same neurons, and in fact, they grow back in the dendrites in a somewhat different way. So it's not rolling back the clock, it's an adaptation and a resilience. Now, in the lower left, and you can see Nad Hines' lab at Rockefeller produced something called a back trap animal, in which there is a protein on the ribosomal proteins that enables the ribosomes and their messenger RNA to be pulled down, so you can look at the gene expression just in the CA3 neurons. And that is what Jason Gray and others in the lab are doing right now. And just to give you a flavor for the effects, at least in this brain area, of early life stress, disrupting maternal care, you can see the pattern of gene expression that occurs in response to a novel stressor. In the blue, these are unstressed animals, a normal environment, and the red are animals that have experienced this early life stress. And what you see is that the capacity of genes to be either turned on or turned off is markedly limited by this early life stress exposure. Now, this, I think, is only the tip of the iceberg, but just to show even more, these are genes that are affected by glucocorticoids. They have these response element sites, and again, in response to this novel stressor. And you can see first, if you compare control and early life stress, you'll see how much different the gene expression patterns are between those two states. And then if you look at forced swim test, the acute stressor on top of that, you can see the expression in the second bar of after forced swim in the naive animals is quite different from that in the early life stress animals. By the same token, if one looks at a series of genes that are related to epigenetic modifications, many different kinds of modifications, including the modifications of histones, the modifications of methylation and demethylation of the DNA. You can see that on the left of each set of columns, you can see major changes in the expression of these epigenetic enzymes. So Jason Gray and colleagues are in the process of putting this together, but I wanted to give you a glimpse of, at least from animal studies, how profound these effects are. Now, this is something that we already heard about, but just to remind you that lack of verbal stimulation, as often happens in poverty, but also in households where the parents are absorbed and neglectful of their children. Lack of verbal stimulation leads to smaller vocabulary, as in the famous Hart and Risley study, Meaningful Differences. That chaos in the home, whether it's due to ACEs or also living in crowded and ugly environments, leads to elevated obesity, elevated inflammatory tone, and as indicated by poor dental health, increases or decreases the development of downstream control by the prefrontal cortex. And so there's less self-regulation. And that is also one of the consequences of the ACEs, as from the Filiti and Anda studies that you've already heard about. One example of the sensitization is from this study by Greg Miller and Edith Chen, where a harsh family climate, often harsh language and roughness in the home, leads to the emergence of a dose-dependent pro-inflammatory response to stressors in the adolescent. And you can see a dose-response curve. The more family harshness there is, the larger the inflammatory response is to some kind of a severe stressful challenge to an adolescent. And that, of course, then systemically and has consequences for physiology of many different kinds. So if we look then, a brief survey of the literature of what early life affects, whether we're talking about abuse or also poverty, the hippocampus is smaller after early life abuse, in poverty, with so low self-esteem, which often is the result of early life abuse, and there's an increased risk for PTSD associated with that. The amygdala is larger and more active in depression and anxiety disorders in general. There's increased vigilance after ACE, after adverse experiences and neglect, and also increased volume even, a study by Sonia Lupien, if there's a child raised with a depressed mother. And the prefrontal cortex, as I've already said, is impaired by chaos, poverty, low SES, and by the adverse childhood experiences. And this is critically important because self-regulation is the essence of our ability to respond in our world. Back to Neil Halfon, if we look at the experiences that are either negative or positive over the life course, it raises the understandable point that you are, of course, not turning back the clock, but at each life stage there are things that could be done to alter things for the better, and there are things that happen to alter things for worse. And we're far from knowing what these things are. This is what both Jack and Nadine were talking about, with things yet to be discovered and applying things we already know how to do from the standpoint of all of us in terms of improving the quality of our lives. We know the importance of regular physical activity. We also know, with increasing the evidence, that mindfulness-based stress reduction and meditation can have beneficial effects on the brain. This study from Holtzel and Lazar at Mass General on people who had an anxiety disorder who did this and who successfully reduced anxiety appeared to have a smaller amygdala volume. And so that's one example. And then finally, social support and integration. The experience corps, where elderly volunteers, it's sort of like the executive volunteer corps, go back, in this case, into schools, become teachers' aides. They become an effective, they have become friends with their coworkers. They become more active, and they are doing something that has meaning and purpose. And I think we underestimate the importance of meaning and purpose as a factor promoting positive health. The whole idea of back to this allostatic load and overload, we have the drains and the gains, the positive, the negative, and the positive, things that are obvious: mental drains, physical drains on the left, and positive gains on the right, both mental and physical, and obviously they interact with each other. This is sort of where we're at. And of course, this has to be projected at different stages of the life course in terms of what it can be effectively done. So looking to the future, obviously brain plasticity, as Jack noted as one of the objectives of the JPB network, it offers many possibilities. We don't know yet how to use it, but the important thing also for those of us, all of us living in this rather hectic life, is to bring back the phrase of Walter Cannon, the wisdom of the body. And fortunately, the body is wiser than we are, and we need to give it a chance to do its job. Thank you.